Transcription is the process by which DNA is copied or transcribed to make mRNA, or the process of synthesizing mRNA. This transcription is the first step of gene expression. What is the difference between mRNA and DNA? mRNA is much smaller molecule than DNA, and it is single-stranded. mRNA contains the base uracil instead of thiamine. The sugar in the nucleotides of mRNA is ribosome, not deoxyribosome. The triplet of bases in mRNA that code for amino acids are called codons. In eukaryotic cells, transcription takes place within the nucleus in the following ways. The enzyme DNA-dependent RNA polymerase, transcription factor, and transcription factor activator binds with a section of DNA, what we call promoter sequence, next to the gene to be transcribed. Then transcription factors activate the enzyme RNA polymerase. After this enzyme gets activated, it begins to unwind a section of DNA. Then RNA polymerase moves along the antisense or non-coding strands of DNA, using it as a template to synthesize mRNA. RNA polymerase moves along antisense strands of DNA using it as a template means it makes mRNA that's complementary with template strands of DNA and therefore identical with sense strands of DNA based on base pairing rule. RNA polymerase assembles free RNA nucleotides to make RNA transcript. The completed mRNA molecule leaves the DNA. In this case, the mRNA molecule now contains the code for protein. Lastly, the strands of DNA rejoin and recoil. mRNA processing. After transcription begins, a methylated GTC cap or 5 cap, or sometimes called guanine cap, must be added to the 5' end of mRNA strand. 3' poly A tail that is supposed to contain 50 to 500 threonine adenine nucleotides must be added by poly A polymerase to the 3' tail. Proteins are also attached to the tail. So mRNA processing help to export mRNA from the nucleus to the cytoplasm. This protects mRNA from enzyme degradation and helps to attach the ribosomes. The last process of transcription in eukaryotic cells is mRNA splicing. We know that in eukaryotic cells, DNA strands contain many non-coding sequences or regions. Therefore, pre-mRNA contains the non-coding regions since it is identical with the gene or sense strands of DNA. In this case, the coding regions on pre-mRNA are called exon and the non-coding sequences are introns. So introns must be cut out and removed, and exons must be spliced together to form host mRNA that purely contain the coding sequences. Let us see transcriptional principles. Codons are identical to DNA triplet codes, except uracil is substituted for thiamine. To form mRNA when transcription takes place, only the antisense strands of DNA is transcribed. Since mRNA is required to be identical with sense strands of DNA that contains the gene. Therefore, mRNA is identical with sense strand of DNA and complementary with nonsense strand of DNA. Translation is the process in which mRNA is transported to the cytoplasm where it is decoded or translated to produce the correct order of amino acids in a protein. Translation takes place in cytoplasm at organelle ribosome. I think you have an idea about ribosome in grade 11, chapter 4. Ribosome is an organelle where protein synthesis takes place. This ribosome can be 80S and 70S. 80S types of ribosome exist within eukaryotic cells and 70S exist in prokaryotic cells. Ribosome is made from ribosomal RNA and protein molecules. Protein molecules make the larger subunit and smaller subunit parts of ribosome. The larger subunit is parts of ribosome where amino acids are joined together to produce polypeptide or protein, whereas smaller subunit is the second part of ribosome where mRNA get read. To join amino acids, larger subunits of ribosome has three sites that can be occupied by transfer RNA molecule. That can be A site or amino acyl transfer RNA binding site, P site or peptidyl transfer RNA binding site, and E site or exit site. Schematic model showing binding sites and the two subunits of ribosome. A site holds amino acid to be added, P site holds growing polypeptide chain, and E site is a site where transfer RNA exit or leave ribosome. This one is a smaller subunit that's responsible to read mRNA, which has its own mRNA binding site. This one is a larger subunit where amino acid containing transfer RNA binds on. All transfer RNA molecules have the same basic structure, what we call cloverleaf structure. The cloverleaf configuration of the molecules, as we can see from the diagram, has anticodon and amino acid attachment site. Anticodon will be complementary to one of the mRNA codon. The following events take place during translation. The first two codons of mRNA must enter the ribosome. Transfer RNA molecules with amino acids attached on its back that have complementary anticodons to the first two codons of mRNA bind to those codons. A peptide bond formed between the amino acids carried by two transfer RNA molecules and the dipeptide is transferred to the transfer RNA in the P site. The ribosome moves along mRNA by one codon, bringing the third codon into the ribosome. At the same time, the free transfer RNA exits or leaves the ribosome, and transfer RNA with the dipeptide moves into the P site. A transfer RNA with a complementary anticodon binds with the third codon, bringing its amino acid into position next to the second amino acid. A peptide bond forms between the second and third amino acids. The ribosome moves along the mRNA by one codon, bringing the fourth mRNA codon into the ribosome, and the whole process is repeated until stop codon is in position and translation ceased or stopped. Protein synthesis in prokaryotic and eukaryotic cells. The process is essentially similar in both types of cells, with DNA being transcribed to mRNA and then translated to polypeptide chain. However, there are some differences in between them. Prokaryotes do not have nucleus. Prokaryotic mRNA does not need post-transcriptional processing or modification because prokaryotic DNA do not contain non-coding segments or sequences. Transcription and translation are coupled. That means mRNA can be translated by ribosomes at one end while it is still being transcribed from DNA at the other end. Both translation and transcription occur in cytoplasm since prokaryotic cells do not have nucleus, whereas in eukaryotic cells, transcription and translation are separated. That means Transcription takes place in the nucleus and translation occurs in cytoplasm. mRNAs need post-transcriptional processing and modification before leaving the nucleus because DNA of prokaryotic cells contain non-coding segments or regions. Some of the proteins that our body synthesizes include structural proteins like collagen and keratin, enzyme like ATP synthase and DNA helicase, peptide hormone that include insulin, adrenaline, antigen that includes antigen A on red blood cells and CD4 on CD4 lymphocytes, antibodies like anti-A antibodies, anti-B antibodies, and HIV antibodies. What are the main sources of amino acids that our body uses to make protein? The main sources of amino acids are protein-containing foods and transamination. Protein-containing foods can be sourced from animals like meat, fish, poultry, egg, and milk. Protein-containing foods can also be sourced from plants, include quinoa, buckwheat, hemp seed, amaranth, cereals like wheat, rice, maize, nuts, and pulses. The second main sources of amino acids to our protein is transamination. Transamination is the process of synthesizing non-essential or necessary amino acids from essential amino acids, or transamination is the process in which amino groups of amino acids transferred to keto acids to generate the amino acid version of keto acid, what we call non-essential or necessary amino acids. Our body needs 20 different amino acids to grow and function properly. These 20 different amino acids can be categorized as essential and non-essential amino acids. Essential amino acids cannot be made by our body and must be obtained through our diet. These essential amino acids are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Necessary amino acids are amino acids that can be synthesized within our body or derived in the body from essential amino acids. These necessary amino acids are alanine, asparagine, aspartate, cysteine, glutamic acid, glutamine, glycine, proline, serine, arginine, and tyrosine.